welcome to today's session on the webinar organized by the World Potato Congress. And while you're just setting up your systems, it gives me the nice opportunity to remind you on the World Potato Congress that will take in Ireland next year in 2022 in the period of 30th of May to the 2nd of June. And yeah, if you're interested to join the World Potato Congress next year, also is anticipating that we are behind the COVID crisis, then you're most welcome to attend, and I invite you to go to the website and to register you for this event. Well, for today we will talk about late blight. We will talk about Phytophthora Infestans. And for today's session, yeah, I've given it a title to say it's about late blight, 175 years of experience, and what have we learned, and it's just an update. So how we are going to do this, what I would like to do is first, let's introduce you to Phytophthora Infestans, to get familiar with Phytophthora. Who are you? And based on that one, we will talk about the frame of integrated control strategy. We'll talk about crop management and primary inoculum, variety resistance, fungicide strategies and resistance management, smart control with digital solutions, and finally, we finish off with an outlook. So first, let's have a look to the historical highlights. It's this year exactly 175 years ago that we had this enormous outbreak of late blight in Ireland. And we all know the consequences. More than 1 million people lost their lives, and another 1 million people left Ireland to look for better opportunities. In this period of time, we had no clue what was causing the destruction of the crop. There were two people that were very much in the research in clarifying. This was very much about Miles Joseph Berkeley in the UK. He published his first findings in the Botanical and Physiological on the potato murrain. And later on, it took a couple of years to really clarify that the causal agent was of the late blight was caused by a fungus. Now, this fungus was described by Anton de Barry. It's a German plant pathologist. And he called this fungus after the impacts on the crop. He called it the plant destroyer. And plant destroyer in Greek is very much the name Phytophthora. So that's where the name is coming from, Phytophthora Infestans, the plant destroyer. Now, let's have a look to Phytophthora. And all of you on the call today, it will be very easy for you that are working with Phytophthora. You know, it's not difficult to find Phytophthora Infestans in the fields, either by infected stems or tips in commercial fields, since it's very easy to detect. If you look to Phytophthora, it is continuous adapting. The symptoms might be a little bit different. Most of the time, we do see the tips, as in the picture on the left. Over the last years, we do see the Phytophthora many more times. It is also infecting the young stems, so it's much more hidden in the crop. And the leaf symptoms with the brown necrosis on the top is very familiar to you. And also we know that if Phytophthora is striking in the crop, the defoliation can go that quick that within 1 week you lose the overall crop. Now, to remind ourselves, we talk about early blight, late blight. So Phytophthora is very much associated to late blight. The other key fungus in potatoes is called the early blight, caused by Alternaria solani. And Alternaria solani, it's different in the symptoms. And what is very obvious, if you look to the symptoms, what is missing is the lack of the white spores on the underside of the leaves. Very typical for Alternaria are these concentric rings. And if you look to the symptoms of these spots, Alternaria, in contrast to Phytophthora, it's much more starts on the old leaves that are weakened either by the environment and or nutrient stress. Now, let's go back to Phytophthora. And to know and to get to understand Phytophthora, let's look into the life cycle of the Phytophthora. And what I would like to do, I would invite you to follow me in the picture. And we start off here with the spores. These spores, as being mentioned here in this picture, they appear on the underside of the leaf. And actually, these spores are the same as what you see the white color here on the leaves. So what is happening with those spores? Well, those spores are being built by Phytophthora overnight. And when morning comes, when temperature or humidity drops, the spores are lifted in the air and the spores are being dispersed by the wind and land on the leaf. Spores under high temperature and what we call direct germination, the spores germinate on the leaves, invade the stomata, go into the leaf, colonize the leaf, and then after about, you could say, 7 till 10 days, you have a new sporangiophores and the new spores, and the cycle starts again. Well, Phytophthora is able to fly, we know, but it's even smarter. Apart from the ability to fly, it is also able to swim. And that means when there is sufficient water available on the leaf surface, then the Phytophthora, it is not germinating directly, but we call it the indirect germination. The Phytophthora forms these so-called zoospores. Those zoospores swim on the water that's on the leaf surface. It swims to the stomata, and then it infects the stomata after the enlistment. In the leaf, you get the colonization, the growth, and once again, the formation of the new sporangia. And the same thing, this appears around 7 till 10 days after landing of the spores on the leaves. Well, the direct germination takes place very much at the high temperatures. And if we say high temperatures, then normally we talk about temperatures higher than 25 centigrade. The indirect germination, you could say roughly it is taking place below 25 centigrades under conditions where sufficient water is being available. Well, once the Phytophthora is not in the plant, we do know that Phytophthora survives in the tuber of the potato, and the tuber is more or less the carrying vehicle for bringing the Phytophthora into the new growing season. Now, what is rather new for Phytophthora is very much this part of the cycle, and is what we call the sexual reproduction. What is happening during the sexual reproduction is that Phytophthora in the leaves of the potato plant, the different types of the Phytophthora they meet, and that means that the fungal mycelia starts to merge, and there is the formation of an oospore. The formation of the oospore takes place after the exchange of genetic material, and that's what we call the sexual reproduction. These oospores are being formed in the leaves. They stay in the leaves and they stay in the fields. And after the leaves are being decomposed in the soil, these oospores stay in the soil, and they enable Phytophthora to infect the potato crop in the absence of the potato plant. So we talked about the sexual recombination, and this is very much related to the Phytophthora Infestans, the concept of the A1 and the A2 mating type. So let's have a closer look what that means. And there are three situations, you could say. You have a situation where you have an A1 and A1 population. It means you have different isolates, but all the isolates belong to the mating type A1. So what is ongoing? You have a mixed population with all type of A1 isolates. There's no exchange of genetic information, and it's just an accumulation of small mutations. That means that over time you have different strains to develop, and the survival is through production of airborne sporangia. Well, if you look to the situation with the A2 and the A2 population, if you have this situation, it's similar. You have a competition for the fittest isolate of the A2 isolates. It is a mixed population. There's no exchange of genetic information. There is an accumulation of small mutation that is leading to the emergence of new isolates, and the survival is still through production of airborne sporangia. Now, the big change comes when A1 and A2 mating types are present in the same field in the same plant. What is happening? You get offspring based on the exchange of genetic information, and the offspring is either an A1 or an A2 mating type. And having the exchange of the genetic information, you get hybrids and you get new strains with new properties. New properties, and we will talk about later on, that makes Phytophthora different in aggressiveness. And what is very important, these oospores are able to survive in the soil. That means if you have a mixed population of A1 and A2 mating type, Phytophthora is able to survive in the soil either through the sporangia or through oospores. Well, this cycle, this sexual recombination of Phytophthora, this is rather new. It is new, and we know this since the mid-50s of the last century. So if you talk about plant pathology, then it's really a recent finding. If you look now into what are the consequences, well, you could say if you have Phytophthora population with a mixed A1 and A2 population, this gives the option for the Phytophthora to unlock the genetic variation. And if you look back into time and in the history, and we will do in a minute, then the regions with native population of Phytophthora in Latin America, such as Mexico, they hold mixed populations, so A1 and A2 mating types. And if you look into the rest of the world remained fairly stable. It's around the mid-90s with the dominance of the A1 mating type. But what is now the difference? Since you could say the early 1980s, a global spread of the A2 mating type took place, and this was associated with the emergence of new clones and the failures in disease control. So, and if you go back into history, it happened in the US somewhere around the year 1997, and this was very much related to the emergence of the isolated US 8 A2. In Europe, it was 10 years later, and in Europe, it was very much related to the clone isolate called EU13A2, called also the Blue 13. And if you look to Asia and Africa, the same happens, but then a little bit later, Asia in time period 2010-2015, and in Africa, you could say roughly from 2015 onwards. So if you look at what has happened over time, then the question is, why are the new Phytophthora isolates so successful, and why are they so challenging to control? Well, this is a simple question, but it has multiple factors to answer. And first, we look to these new Phytophthora isolates, and we look to the expanded host plant range. So as you know, Phytophthora likes very much potatoes, and beyond potatoes, also tomatoes. So for a long time before that, potato and tomato would be the only host for Phytophthora. Meanwhile, we know that in the Solanum family, also other Solanum weed species act as an alternative host for Phytophthora. And on the slides, as I mentioned here, many different weed species are mentioned. And I just draw your attention to the hairy nightshade. The hairy nightshade, this is a weed species that has a prevalence in Europe, but also in the US. And the research has indicated that the hairy nightshade is a very effective alternative host for Phytophthora. And that means that in controlling Phytophthora, that weed management gets importance. So that's about the expanded host range. If you look to the features of Phytophthora, and it's also about the increased aggressiveness. We have mentioned before that the sexual recombination took place and you get the formation of the oospores. Along with the formation of the oospores, we have higher genetic variability in the Phytophthora. And that means there is an ongoing selection of aggressive strains. And these ongoing selection with new aggressive strains, they have a shorter incubation time. They have an increased sporulation intensity, an increased infection efficiency, increased stem and tuber infection, and there is also an increased growth even at lower temperatures. Well, if you look into this list, then the shorter incubation time, that is certainly a topic to keep in mind, since we know now that in today's situation, the most aggressive strains, they have an incubation time, meaning the time between spore landing and the time that new spores appear. In the current situation, this is around 3-5 days, whereas in the old situation, it used to be in between 7 and 10 days. So this is about the increased aggressiveness. If you talk about the start of the epidemics, around the world, we do see that there is an earlier start of blight epidemics. The question is why this is. Well, it is for sure related to climatic change. It is warming up earlier, but it's also likely an adaptation of the Phytophthora to lower temperatures. And if you look to the primary inoculum sources, then it's important to keep in mind that the primary inoculum sources is very much about infected fields in warm regions. And very much if you talk about regions like Asia and Latin, where you have continuous cropping conditions, then the infected fields are a very important source for the early start of the epidemics. And the other one is, we should not forget, is about infected tubers. And if we say infected tubers, then there are different routes for infected tubers. It is about the potato dumps, outgraded piles, and as being mentioned here in the picture on the right. So these are the potatoes that are not suitable for consumption or for planting. And these left potatoes, they are a source for development of potato plants not being protected and producing a lot of spores that move into the new potato fields. The other topic on infected tubers is about volunteer potatoes. These are potatoes that remain in the field and emerge in the succeeding crops, and once again, not being protected, producing spores. And the last one is about seed potatoes being infected with Phytophthora. The early start of the blight epidemics is also associated to the oospores. This is illustrated with the picture here on the bottom. It means that the Phytophthora, it starts coming from the soil, and certainly on the conditions where we have heavy rain, intense showers, then we see the splash spreading of the Phytophthora coming from the soil, and Phytophthora being moving from the soil directly in the leaves of the potato plant. And what we have mentioned before, the earlier start of the epidemic is also related to widening of the host plant range, meaning that the Phytophthora is emerging from weeds in the rotation. Yeah, and if it is not enough, then there is an increased virulence on potato cultivars. So if you look to the new isolates, it is known now very well that the new isolates are much more virulent on the current potato varieties. Well, how to explain that one? This is a bit a complicated slide. So I would suggest we first look into the first picture on the top. So what do we see? Well, we see here different plants, and different plants have been used. And just as a backup, each plant is containing a different resistant genes. So starting from the left, we start off with a plant containing resistant gene R1, then the R2, and so we follow to the right till the plant with the resistant gene R11. What has been done? These plants have been infected with a Phytophthora isolate called the EU8A1. And if you spray the plants with this isolate and you assess them 1 week after, then this is the result what we see on the picture. And if you look into the picture, what we do see that some resistant genes are not effective in controlling the Phytophthora. This is the case for the resistant gene in the plant R1. The same is for the R4, and we see the same for the R11. Well, the good news in this situation on controlling the EU8A1, that there are a couple of resistant genes that are still very effective, like the R2, the R5, the R7, and even the R8. Now moving to the picture down, it is the same procedure. The only difference is in this case we have inoculated the plants with the virulent EU13A2 isolate, which used to be dominant in Europe over the last years. If you look now into the reaction of the plants, you do see that out of all those resistant genes, there are actually two resistant genes standing out, is R5 and the R8. And the rest of the resistant genes, they have lost their efficacy in controlling Phytophthora. And that is one of the observations that growers made, that the cultivars currently used in practice, they are not able to prevent the spread of Phytophthora, and these cultivars are not able to control the Phytophthora, linked to loss of resistant genes in controlling the Phytophthora. How do we know all this? I would say, well, we know this since we started doing the Phytophthora monitoring. And the Phytophthora monitoring and the improvement in monitoring was enabled by the DNA fingerprint technology. So how does it work? It means that scouts issue with sample forms and what we call the FTE cards. These cards, scouts, they note and record the outbreak data. That means the location, the crop type, and the cultivar. And once being in the field, these scouts, they press the lesions of the Phytophthora in the leaves into these FTE cards to capture the pathogen DNA. Then this DNA of the Phytophthora is fingerprinted using 12-plex markers, what we call SSR markers. And having these 12 different markers, we are able to identify the different isolates. Then these genotypes, they are defined, the data are stored, in this case in the EuroBlight database, and this EuroBlight database is available to advisors, to growers and researchers to learn on what is the actual situation in the genetic makeup of the overall potato population, Phytophthora population in a given region. So how does it look like? Well, very colorful. This is a slide. This is already dating back to the year 2008, work done by the James Hutton Institute. And the credits also go very much to the James Hutton Institute that has developed this method. Taking a look now to this chart, what do we learn from that one? Well, first we could say it's very colorful. Yes, it is very colorful. But how to read it? Well, it means if you start on the left with the year 1982, then it means each color in this graph represents a different genotype. So in the year 1982, you could roughly say there are 4 major genotypes, of which this light blue color was the dominant one. If we look now over time and we move from the left and we move to the right and to the year 2008, then we see differences happening. Different colors are popping up. In 2003, we had the red color dominating. And if you look to the year 2008, we see that the blue color is coming up. Well, the color in this graph has no meaning. It was just mentioned before that is very much the man that made these graphs. He chose the color to have a clear differentiation. Coincidentally, the color blue was associated to a very dominant genotype, and later on, many of you have heard the name before, blue 13. Well, this is very much linked to this graph, blue 13, associated to the EU 13A2 isolate. This is the situation in the UK, the year 2008. Work has progressed since, and the work out of as being initiated in the UK has been implemented through the Blight network throughout region Europe. This is a graph very recently coming out of March 2021. It also includes the results and the actual status of the year 2020. If you look to the picture again and as a graph, we see it's very colorful. Yes, again, it's very colorful. We also see that the blue 13, as what was mentioned before in the UK, it used to have a high dominance in the Phytophthora population in Europe. But ever since farmers adapted their spray program and started to reduce the amount of metalaxyl-based products, the selection pressure for this specific isolate, the EU 13, went down, and consequently we see now that the blue 13 is not that anymore important in the European Phytophthora population. At the same time, if you look in Europe, selection is still ongoing. It's still a survival of the fittest of the new isolates. And meanwhile, in Europe, we see that there's a dominance of A2 mating types. Well, this is about Europe. We could do the same analysis for APAC. And if we look into APAC and the work that has been done in APAC, then we could say, well, in Asian Pacific, if you go back in history, Asia Pacific, the Phytophthora was very much dominated by the isolate we call the FAM A1. Afterwards, till about the 50s, you could say this one was replaced by the US number 1, also an A1 mating type. And if you go into the 80s of the last century, then we see that the first A2 mating types are arriving in Asia Pacific. And if you look back to the outcome of the year 2020, under the current conditions in Asia Pacific, the dominance in the population is very much related to isolates that belong to the A2 13 family. It's interesting, selection pressure is ongoing. The reason for this selection, that is a good question, and this is what researchers have not agreed upon. What are the key drivers that this selection pressure is taking place? Though in Asia Pacific, we do know there is an overuse of metalaxyl, all type of metalaxyl products, and the overuse of metalaxyl containing products is selecting for the A213 family under the conditions in Asia Pacific. Well, this is about Asia Pacific. Let's now move to the US. The US also changes are ongoing, but once the drivers and the history is different compared to what we have seen in either in APAC or in Europe. Going back in history, the US very much similar to Europe and APAC. Original, the FAM A1 was dominating, followed up by the US 1, also an A1 mating type. And what we do see in the US, what happened is in the mid-90s, that there was a disruption in the Phytophthora genetics in the US with the arrival of the A2 mating type, the clone A28. And the clone A28, it was an isolate that has a high resistance to metalaxyl, and consequently, at that time when farmers were not knowing that metalaxyl was selecting for this isolate, the overuse of metalaxyl caused the rapid spread of the A28 under the American conditions. Well, we learned over time, we reduced the metalaxyl intensity in the US, and we moved into a situation where we introduced another A2 mating type, and it was around the year 2010. Very interesting, this one originates from tomatoes. If you're talking about potatoes, it's always good to keep in mind what is happening in the other members of the Solano family. So in 2010, the A222 was introduced in the US, and if we look to the recent data in the US, very interesting, the A2 is almost absent in the US, and the overall population in the US market is very much dominated by the A123 clone. And interesting, this clone is aggressive on both potatoes and tomatoes. Yeah, and then if we conclude, then we could say, what have we learned? What have we learned? Well, there is one big learning, and the one big learning together, it is we underestimated genetic potential of Phytophthora. We underestimated the ability of the Phytophthora to adapt to new growing conditions. That means lower temperatures, lower relative humidity, with a shorter life cycle. And the shorter life cycle, as mentioned before, the incubation time is compared to the past, it's currently 3 till 5 days compared to what it has been 7 till 10 days in the past. The Phytophthora has been able to overcome major resistant genes. It has been able to develop fungicide resistance, and this is mainly associated to the products related to metalaxyl and mefenoxam. And unfortunate, if you look now in controlling Phytophthora, unfortunately, the long lead times and innovation cycles in both potato breeding and in the crop protection industry is reduce our options to effectively intervene and control Phytophthora. So what do we learn with that one? It means for the future, we need an IPM, an integrated pest management strategy to control late blight. And by doing so, we need a holistic approach. If you talk about this holistic approach, then we talk about a control strategy that is based, you could say, on 6 factors. It is about crop management. It is about monitoring of Phytophthora populations. It is about the use of resistant cultivars with different resistant genes. It's about implementing a fungicide strategy according to crop characteristics, and that's very much about the R gene content and the crop stage and weather conditions and disease pressure. It is about implementing a resistance management strategy for fungicides, and it's to adopt digital technologies to enable smart control strategies. Well, in the next slide, I will guide you through, and I'll guide you through the overall strategy. Let me start off with the primary inoculum. And on primary inoculum, it is always good to remind ourselves what is primary inoculum. On this picture, you see the different sources. On the picture on the left, you see volunteer plants. These are potato plants in a succeeding crop. The sugar beet, this is a picture taken in Europe, where sugar beet are grown after potatoes. And when the winters are mild, the tubers don't get frozen, and those potatoes, that's tubers that survive in the soil, emerge in the succeeding crop. If you look into the picture on the top right, then you see a picture that's coming from Africa. And in Africa, it is quite common practice that farmers use intercropping. And it means, in this case, corn with an intercrop of potatoes. It means in principle that you have more or less a continuous cropping of potatoes along the rotation. That means an easy survival for Phytophthora to jump from one crop to the other one. And the last topic is about the potato dumps. This is a picture also taken from Europe. These are potatoes that are not suitable for consumption or for sale. They are left behind the barn or somewhere in the field. Potato plants that are developing on these potato dumps are not protected. They produce spores, and those spores move into the new potato fields, causing a late blight. So if you talk then about crop management, and we want to control primary inoculum, then the reason why we are doing this, it is very much to prevent continuous green bridges and to break up those green bridges between either the potato crops and or potato and tomato crops. To start off, and this is very much a general concept about the crop rotation, it would be a very good practice, easy to say on the slide, to have a crop rotation in with potatoes with around 30%. What does that one mean, 30%? It means 1 out of 3 years potatoes on the same field. Well, I can make this claim very easy on this slide. I do know on the practical condition it's much harder to implement. But keep this in mind, it would be a very good rule to reduce Phytophthora, not only Phytophthora, but also soil diseases, to have a rotation in 1 in 3. If you talk about reduction of potato volunteers in the rotation, there are different ways to do it. The modern harvesters, machine harvesters, they have now what they call the squashers on a machine. They are mounted on a machine. That means that the small potatoes, which are not being picked by the harvester, they are being squashed on a machine, and they do not germinate after the harvest. Also, a very effective measurement to control potato volunteers is the application of anti-sprouting agents before harvest. And the maleic hydrazide is a very good compound for that one. And it means that those potatoes that are left in the field, it will not germinate, and consequently, they will not support the onset of Phytophthora in the next season. Last topic to mention, volunteer weeding, absolutely important to remove the potato plants in the succeeding crops. Talking about the control of the outgraded potatoes, well, I brought here a picture, and this is the picture here down under in the right. And this is a simple way. So for farmers, it's a very simple and cost-effective way to make sure that Phytophthora is not escaping the potato dumps. That is just to cover it with black polyethylene plastic. Well, if I look into the Europe, there is an agreement between growers to make that one happening. Talking about a country like the Netherlands, they even have the grower association together with the plant phytosanitary services. They agreed on the principle that when those potato dumps are covered before the 15th of April, it's all fine, it's green. However, when it is not being covered after the 15th of April, then first you could say, as in the football, you get a yellow card, to say a warning. But this warning comes with a red card. That means a big fine. And what is the fine? The fine is 500 euros a day after the 15th of April for not protecting the potato dumps. And meanwhile, farmers are very happy with this concept. It is a community approach to support each other to reduce the impact of Phytophthora. Going back now to the tropics, I've mentioned the reduction of the intercropping. That is a very attractive way to do it, but more work is being needed to create awareness for the farmers and also to bring the tools for the farmers to make sure that they have an income once they change their cropping system. Weed control, we mentioned it already before, ensure effective weed control across the rotation and very much for the other Solana species. And we mentioned here the hairy nightshade as an example. Yeah, and then coming back on the crop rotation, once we have a good crop rotation, 1 out of 3, we will have an effective way to reduce the impact of oospores as a source for early start of the Phytophthora. Since oospores, they could survive for 4 years, but in general, we state that oospore survival beyond 3 years is very low. Then we move to the cultivars, adoption of resistant cultivars with resistant genes. And what the intention is very much to grow potatoes with different resistant genes and multiple resistant genes. If you look into the current situation, that the majority of the current potato cultivars, they hold resistant genes that are not effective anymore and that lost their protection versus late blight. The best example of that one is when we go to the US, and there we have the russet Burbank variety. The variety is meanwhile more than 100 years old, and there are no resistant genes available in russet that give protection versus Phytophthora. Well, there's good news. There is good news, and the basic research over the last year generated a lot of insight in the Phytophthora resistant genes. The resistant genes have been identified, the location on the chromosomes have been identified, and even the mode of action have been identified in the so-called small spectrum and broad spectrum resistant genes. So that means there is a lot of new insight. What is though important is to understand that the mode of action of all those resistant genes is based upon the hypersensitivity response. And that means there's a programmed cell death in the potato leaf upon the recognition of the Phytophthora by the plant. Since remember, Phytophthora is only to grow in living cells. So as fast as the cells are dying, the harder it is for Phytophthora to survive. So, and what is more positive, that multiple new resistant genes will become available for the new potato varieties over time. Having a little closer look now on those resistant genes, and this is a slide I plugged into the presentation, is based on the work done by Wageningen Research. In this case, is Jacques Vossen. And he made a very nice overview on where all those different resistant genes are being located in the genome of the potato plant. So this is very good news for the breeders also, that we exactly know what we are targeting, and with the market-assisted breeding, there's a very efficient way to bring the resistant genes into the new cultivars. So far, we have roughly 20 different resistant genes available, but I would say there it starts the good news. Since partly of the good news is being tempered, having the additional research on the mode of actions, meanwhile, we understand that although we have 20 different resistant genes, if you look into the mode of action, we come down to 10. And those 10 resistant genes with different mode of action, they are mentioned here on the right of the slide. Well, that brings me to the next slide, and that means if we are going to grow potato cultivars with resistant genes, then we have to be very careful in the adoption. That means careful in the adoptions, and even though 10 different mode of actions have been identified, late blight control is effectively based on 7 resistant genes. And why this is, it is that out of the 10 resistant genes that are available, different mode of actions, 3 of them lost already efficacy in controlling Phytophthora. So for the new varieties in controlling Phytophthora, we are relying on 7 broad spectrum resistant genes that provide effective control of late blight. And those 7 major resistant genes I've just mentioned here with the shortcuts. How are these resistant genes now coming available? Well, they come available through new breeding techniques. It's all about non-GM, so it's very much about the classical breeding techniques with market-assisted breeding. Also, the The true potato seed, the haploid, or let's say the diploid hybrid potatoes, they accelerate the speed in introducing varieties with one or multiple resistant genes. And very much as we call it more resistant genes, we call it the stacked approach. Now, what is very important for the overall industry is that we have transparency, and careful management of those resistant genes employment to make sure that we implement a sustainable and durable late blight resistance concept. And for that one, potentially, I would say it is an option for the breeding colleagues to share this information on the mode of action of the resistant genes, similar at what the crop protection industry is doing for fungicide resistance. The principles are very much the same. And potentially the guidelines that set forward and the method used in the FRAC group could be a reference also for the employment of the resistant genes in the potato plants. Yeah, and then we come to the fungicide performance. We didn't talk about it, but when we are not able to control Phytophthora through resistance, then our last hope is the use of fungicides. And fungicide performance is very much having a double challenge, since in applying a fungicide, we need to make sure that we both control the foliar blight and the tuber blight. And as you may recall, the spores being formed on the leaves, they are being washed down by the water. It's either rain, irrigation, or dew. These spores move down into the soil, and once the spores come in contact with the tubers, they are able to infect and to colonize the tuber and to destroy this tuber either in the field or during storage. So it is very important that we have a fungicide program that has this double challenge in an effective control of both foliar and the tuber blight. Now, talking about fungicide control and knowing the Phytophthora, it's unfortunate, but we have to secure foliar and tuber blight control from the very beginning of the season till the end. And if we look into what is available, we have contact and translaminar fungicides available. The contact fungicide you know very well, and if you talk about contact such as a mancozeb product that is well known around the world as an effective product in controlling late blight. And then there are different fungicides with translaminar activities. We come back to that one a little bit later. All what we do, the fungicide choice, this should be directed by crop stage, by weather, and by the conditions, and by the disease pressure. And very important for an effective control strategy, ensure that the spray application and the intervals are adjusted and in such a way that it covers the protection of new growth. Protection of new growth and successful protection of new growth is the basis for a very effective control strategy for late blight. Yeah, what I mentioned before on the resistant genes used in the potatoes, it is very much applicable for fungicides. We need alternation of fungicides with different mode of action to ensure active resistance management. And this topic I will clarify later on in the table to share with you the different mode of action and what can we do to implement an effective strategy. A little bit more about tuber blight. Also in tuber blight, we learned a lot over the last 20 years. Whereas we thought in former years that tuber blight is a thing that you only do at the end of the season and just control end of the season. Meanwhile, we know that in controlling a tuber blight, it starts much earlier. If you talk about a tuber blight, we know that once spores are arriving in the soil, these sporangia are able to survive in the soil. And the sporangia in the soil, they survive, depending on the conditions, you could say in between 15 and 80 days. But if you look on the commercial conditions, it means once tuber initiation has taken place, it means once tubers are being available, tubers are sensitive for an attack by late blight sporangia. So therefore, it's so important to keep Phytophthora out of the crop. Talking now about fungicide performance, if you talk about fungicide performance, yeah, the performance relies on multiple interactions. It's about interaction with both the plant and the late blight fungus. And if you talk about the performance, many times we talk about what we call bioavailability. It is the bioavailability of the active principle in the fungicide that makes that the fungicide works. So it's the bioavailability and the uptake in the plant tissue. Talking about, you could say the availability in plant tissue, the contact fungicides such as mancozeb, they have properties in sticking properties. So they stick onto the leaf surface and they don't move into the leaves. And the sticking properties give them a good basis to control the germination of the spores of Phytophthora. Whereas if we have translaminar and systemic fungicides, They have both properties. They do and stick onto the leaves to stop the germination of the spores, and they have the properties to partly move into the leaf tissue. And then the last topic, what is important about the activity of fungicide is the stability of the fungicide in and on the plant tissue to guarantee the longevity of the protection. Associated with fungicide performance is the ability to control foliar and tuber blight. Unfortunately, this is not straight ahead. It means that in our current portfolio, the majority of the product is able to control foliar blight. It's unfortunate that we only have a very limited of fungicides available that are also able to control tuber blight. So that's also an area of concern if you talk about resistance management, that we protect those actives that are active on tuber blight, so we do not have that multiple mode of actions available. Talking about mode of action with respect to resistance management, in general, we use the two classifications in fungicides. We talk about multi-site and single-site fungicides. Multi-site fungicides such as mancozeb, chlorothalonil, they have a mode of action that targets multiple metabolic processes in the Phytophthora fungus as a target. And having this multiple metabolic processes that are targeted at the same time, the likelihood to develop resistance or to overcome the activity of fungicides is very low. So we do not have examples that Phytophthora has been able to develop resistance to mancozeb or chlorothalonil or any of those other multi-sites. So these products are very, very important for overall effective resistance management. And why it is, since the single-site fungicides, they are single site. The word is it's already in the wording, single site, very specific. So they very much inhibit specific metabolic processes as a target in the Phytophthora. Consequently, is they are very high in activity. So the intrinsic activity is very high. Dose rates are many times very low. But the negative is, and this is a negative, and the risk that the Phytophthora is able to overcome the activity of these fungicides and is able to develop resistance. Therefore, resistance management is so important to ensure that we keep the activity of these single-site fungicides available. Yeah, a nice picture in between. I just would like to bring you back to the movement of the fungicides and the availability in the plant. And therefore, this is a picture back from our school books, just to explain the two transport systems we have in the plant. As you're all aware, we have the xylem transport that is responsible for the take-up of the water and the minerals out of the soil through the roots, distributed to the leaves, and it is a one-direction transport from the roots to the leaves. The second one is the phloem transport. This is the transport for sugars and amino acids. It's transport directed from leaf to leaf, or from leaves to the roots, or from the leaves to the top. So this is a bidirectional transport system in the plant. Now, these two transport systems, this is so simple, though they have a big consequence if you talk about the mode of action and what fungicides are doing. So if you do the same picture and we talk about the fungicides, then all the fungicides, they have one limitation. That means penetrant fungicides, they have a mobility in the plant tissue, yes. But the mobility in the plant tissue is restricted to the xylem, and that's the big limitation. So if you look what is happening, these penetrant and translaminar fungicides, they are partly taken up in the xylem, they are transported into the leaves, and what is very important to understand, there is no export out of the leaves. There is no export out of the leaf, there is no redistribution. If you talk about the phloem, what about the phloem? Well, it's very simple. There are no fungicides available today that are able to be transported in the phloem. So the current fungicides are not mobile in the phloem. And this having a big consequence for the potato growth. It means that new growth is not protected through redistribution in the phloem. That's exactly the reason why potato farmers have to get back to the crop so many times in the season. We do not have effective products to control new growth. That means when there is new growth, growers have to get out in the field again to reapply a fungicide to protect the new growth. Yeah, when we talk then about the key learnings in late blight control, when we could say, well, Phytophthora is always present at the start of the growing season, hopefully at a low density, since it's building up, and at the end of the season, normally we have a high density of Phytophthora. It's also good to remind that the fungicide do not kill the Phytophthora, but it prevents the spread. And if you talk about effective control strategies, then it's very important to start preventative control programs before the onset of visible symptoms. to adjust the fungicide choice to crop stage, disease pressure, and weather conditions. Keep the strict fungicide program. Do not compromise on the spray intervals. And this is very important with the consideration of a good protection of new growth, and is also associated to the aggressive nature of the new isolates. Ensure the early application with penetrant products to keep the disease out of the crop, and wherever possible, use the decision support system for a timely, informed intervention. Talking about application technology, not too much time today, just to mention, if you talk about the tractor-mounted sprayers, just make sure sufficient water is being used, and it is very important that we ensure good coverage of the leaves to protect new growth. Yeah, and now I come to a table that I promised you I would come back. This is about resistance management and fungicides. Probably this is the most complicated chart of the overall presentation. It is a table based on the FRAC group. This is the Fungicides Resistance Action Committee. This is an industry corporation, whereas different manufacturers share information about a mode of action and about the results of the monitoring. Well, I've used this table to explain on the farm level what he can do and what he should do. And if you would ask a farmer, and we start in this table here on the right, and you would ask a farmer what he is doing, and you would ask a farmer, do you have a good anti-resistance management strategy in place? It might be that the farmer is answering you, yes, I've used the products, and let's take here the example. I've used the products Acrobat, Fulban, Melody, and Rivers. So I've used 4 different products, so I have an excellent anti-resistance management in place. Well, from farmer perspective, probably we would say, yes, you're right, you're correct. However, if you look into the common name, still on the chemical, would say, yeah, it's still okay, all different names. If you look to the chemical classes, we already see it's coming down to 2. But what is important to understand is in the end the mode of action. And the mode of action of all those 4 products, it is based on the same mode of action, is about cellulose synthetase. And this is so important that if you talk about resistance management and effective resistance management strategy, that farmers, advisors understand that we have to alternate products that are based on a different mode of action. So this is an example that it is not good to alternate products out of the same mode of action. I've plugged here another example of a product, Infinito, where you have a product that has an inbuilt resistance management based on the propamacarp. This is based on the cell membrane permeability. And the other one is through a picolite. It is about the spectrin-like protein. So you have one product having 2 different modes of action. And that's the ideal world, what we would like to do as industry, to provide farmers with mixed products, having inbuilt resistance management, and by doing so, reduce the risk that the resistance is able to develop. So overall conclusion, for effective control of late blight, alternation of fungicides with different mode of actions is absolutely essential to ensure active resistance management. Digital technologies. We didn't discuss too much about digital, but there's a lot about digital technology is available and will become available. So if you look to the decision support systems that have been developed, they are very much developed to forecast late blight infection events. If you look on the employment of these decision support systems, these DSS systems support farmers on optimum timing and the right fungicide choice. And this is very much in the models according to crop stage and to weather conditions. If you look now towards the future, what is happening in the back is about data analytics. It's about the genomics, as we discussed in the Phytophthora and the different clones. It's also about the effectors. So this is very much about the recognition of the potato plant of the different Phytophthora clones. And in the future, these data analytics will support growers to de-risk their operations. So if you look in the time to come, the sophisticated decision support systems that will integrate pathogen, host, and fungicide data for decision rules. And that would mean that we have included the Phytophthora clonal types and the R virulence genes. We will have included the variety resistance, meaning single versus stacked resistant genes, and we have included fungicide sensitivity. And having these sophisticated systems that will enable the farmers to improve their overall control of Phytophthora. So that brings me to the conclusion that if you want to implement this strategy, an integrated IPM strategy for late blight control, then we need support, we need partnerships on global level, and we need much stronger corporations. Well, Actually, this is already in place. If you look to that one, then for the Phytophthora and the late blight, we have excellent networks available on both on global and regional late blight networks. I've mentioned here 4 of them. It's about Euroblight in Europe. It's about US Blight. It's Tison Latino in the LATAM, and it's about Asia Blight in the APAC region. And Africa, it is starting in East Africa to be associated to Euroblight. So these platforms, they are very important, and I invite you to visit those different websites of the working groups. So this is a platform where we have partnerships, and the partnership between research institutions and private industry sector partners, that makes that we have the complementary expertise, the critical mass, and the resources available to effectively bring all the tools together for an integrated pest management control of late blight. And before closing, I would say I would like to acknowledge all the research institutions that provided their support in putting this presentation together. And sorry, I've not included all of them, just the major ones. And I will make one exception to say thank you very much. This is very much to the James Hutton Institute in the UK and the Euroblight group that has been very much, they have been initiators, I would say, for the current global late blight groups. And the last topic is very much thank you to my Bayer research and development colleagues around the globe. And with that, I would all invite you on the talk to say, let's partner and collaborate and to jointly master late blight. And with that, I will conclude the presentation for today. What I would suggest to do, that we have now a look to the questions. So let me look to the questions, and I guess the questions are coming in. And they do. So looking to the questions, I will have a look to the first one. The first one says, what is the best chemical fungicide against late blight? Well, yes, that's an easy question. And sorry that I make the answer a little bit more complicated. So I would say the fungicide choice, the best one is always depending on the conditions. So it depends on your crop stage, it depends on the weather conditions, and it depends on the disease pressure. And exactly this combination of those 3 factors, and I've forgotten then to mention the other one is about the resistance in the potato cultivars. So these factors combined make that you come with a choice for the best fungicide. To give you more guidance, I suggest you reach out to the Euroblight. In the Euroblight network, there is a fungicide table that gives you some guidance on what is the right fungicide for the right conditions to apply.
Albert, may I interrupt? Could you please
Yes.
stop sharing your slide? That way we can see your face in the full screen. Thank you.
Yes. Oh, yes. Yes, I will do. Okay.
Very good. Thank you.
Thank you. Yes. Let me have a look to another question. I'll read it out. This is a long question. It's about, and the question goes as Europe moves towards the goal of reducing highly effective synthetic chemical inputs, what about the options for biocontrol agents? Is this something industry is focusing on? Yes, this is a very important question. In Europe, and if you look into Europe as part of the new policy document, as what we call the European Green Deal, the field to fork strategy, there is a high pressure on the use of chemical products for controlling late blight. It's true. That means also towards the future, many products will lose the registration. And that means there is a need for alternative solutions. One of them is biologics. So the industry is working on these alternative solutions, biologics. Though we have to understand that to find the lead in a biological solution, it is not easy. A lot of work is being implemented already, but we will need time. We will need time to come up with good product concepts that have robust activity, that deliver reliable control for Phytophthora to de-risk the Phytophthora onset for growers. So yeah, activities are ongoing. When is this coming? Well, I would say we have to be patient. That will be beyond the year 2025. Another question to say, okay, yeah. So I read out a question, how much it is likely that the foliage blight transfers to the tubers, Or is it a different spores that cause foliage and tuber blight? Yeah, that's a very interesting question, and thank you for posing that question. The question is how likely it is that spores being formed in the foliage are moving to the tubers. How likely it is? Well, it is very likely. In general, what you need is water. And then the question, how much water you need, either rain, irrigation, or dew. And research has been done, and it indicates that you only need 5 mils, less than 5 mils per square meter, to bring the spores down from the foliage to the tubers. The question also is saying, are the spores different, yes or no? No, it are the same spores. So Phytophthora, I mentioned, it's very smart. It's able to fly. The spores are able to fly, they're able to swim, and they are able to being transported in the water down to the tubers. These are the same spores. The spores are the same. It just depends on where the spores land, on what disease they and problems they cause. So when the spores are germinating on the leaves, you get the spots on the leaves. When the spores brought, same spores being brought to the tubers, you get the infection of the tubers. Another question, are pesticides responsible for selection of novel pathotypes in the blight population? Yeah, this is an interesting question, and I would say I would open it up more broadly. And the question is, what is responsible? What is now responsible for? What are the key drivers that we do see this ongoing selection pressure in Phytophthora? And actually, we don't know. Could let's say the pesticides being responsible? Well, partly yes. So if you look to what I mentioned about the metalaxyl resistance, if you have metalaxyl resistance, as in the A213 family, then for sure, if you apply metalaxyl as a fungicide on these isolates, they will not control, and they will further select these isolates and support the spread of these isolates. So in this situation, yes, there's a very clear link. For the rest of the fungicides, we don't believe there is a link. It is very much related to the natural population of the Phytophthora and the sensitivities in the natural population of the Phytophthora, whether a fungicide will have an effect on the selection pressure, yes or no. And the big difference is that we have seen in the selection pressure, what is happening in the US, where we have now the dominance of the A1 mating type, compared to Europe, where we have a dominance of the A2 mating type. It shows you there are different drivers in place, and much more research is being needed to understand what those key drivers are. Other question that goes to, can you please explain the difference on how fungicides travel in phloem, xylem in the plant, and give examples of fungicides? Yes, this is an important question, and I addressed this in the presentation. And sorry, it went all that fast on the presentation. If you look to that one, the answer on fungicides is say, can you explain how they move in the xylem and the phloem? Well, the answer is very easy. Contact fungicides. We talk about contact fungicides, as in the word contact, they stay on the plant surface, on the contact. They don't move in. If you talk about the translaminar, or what we call penetrant, or you may call them systemic fungicides, they have the ability to move into the leaf tissue, and they have the ability to move into the xylem system. And that means that when they are taken up in the xylem, they are being redistributed in the same leaf. Important to understand, there is no redistribution out of the leaves, and there is no redistribution from leaves to growing tips. What about the transport of fungicides in phloem? Could you give examples? Well, it's very easy. There are no fungicides available that are able to be transported in the phloem of the plant. So the industry is working on that one, and we have to see whether the industry will be successful in the future to deliver on that promise. And with that one, I would suggest that we stop the session for today. I guess we are almost close to 1 hour. Before closing, I would like to thank the World Potato Congress for setting up the infrastructure to make the presentation available today. And thanks to the organizers to give me the opportunity to share the information with you. And for now, I wish all of you a very good season. When it comes to the research community, I wish you very successful trials and successful outcomes that will improve the overall control of late blight to come. And when you are growing potatoes, I wish you a bounty harvest with minimum impacts of Phytophthora, so having a harvest in good quality. and important for all of you in times of COVID. Take care and stay healthy, and looking forward to meet you soon face to face. Thank you.